This episode is brought to you by SkinCeuticals, the #1 medical aesthetic skincare brand worldwide and the leader in antioxidant science. To learn more, visit www.skinceuticals.com.
Hello and welcome to Skincare Confidential, a podcast supporting the Science of Skincare Summit, an annual educational conference happening November 8th through the 10th in 2024 in Austin, Texas. I am Dr. Ted Lane, a co-founder and a co-director of the Science of Skincare Summit. I am also your host for today, but enough about me. We have a very special guest today, all the way from Germany. This is Dr. Jean Krutmann. Dr. Krutmann does incredible research on the effect of the environment on the skin, in particular in skin aging. He is a professor of dermatology and environmental medicine at the Leibniz Research Institute for Environmental Medicine. Dr. Krutmann, welcome and thank you for joining us.
Well, hello. Nice meeting you, Ted, and it's a pleasure being here.
Great. Well, Dr. Krutmann, you have published extensively on this subject. You are quite legendary, quite frankly. I think you actually were one of the seminal scholars on the effect of air pollution and other environmental factors on aging of the skin. So it's really an honor to speak with you today.
My pleasure.
Let's dive into it, Dr. Krutmann. So you recently published some research on the effect of heat and humidity on the skin. Can you just give us a quick overview of what you found?
Yes, actually, this is the initial observation comes from an epidemiological study that we have conducted in India, because at that time we were primarily interested in air pollution. And as probably you and most of the audience know, in comparison to other parts of the world, India is really paradise when it comes to environmental medicine because pollution is very, very bad. And so we had the opportunity to assess 1,500 Indian women. 500 were based in Delhi, which is the world capital of air pollution. 500 in Mumbai, which is still very heavily polluted, and 500 in Bangalore, which at least for Indian standards can be considered moderately polluted. So we had a very nice gradient. And in addition to looking at all these PM 2.5 and other parameters for air pollution, we also assessed climate factors, in particular one which is called the heat index, which is a function of ambient temperature and relative humidity. One of the reasons why we got very interested in this is that there is increasing evidence that global warming and the accompanying increase in ambient temperature is actually worsening the health effects which are caused by air pollution. This is, for example, known for lung inflammation, but also for some of the other effects. So the interesting thing now was that we found that the heat index per se, without any of the other factors, already contributed to the risk of developing skin aging signs in the face. And this was very prominent and significant when one looks at the odds ratios for skin aging phenotypes related to hyperpigmentation. Which in particular in the Indian population is of course of significant relevance. So the studies are not that easy because ultraviolet radiation exposure, of course, is a very, very important factor when it comes to the development of hyperpigmented spots or maculae in the face. And when temperatures go up, then usually you also have more sunshine. So this is the classical example of a confounding effect. So we had to make sure that our statistical models were, first of all, very, very robust and that we could precisely control for any confounding. And the way you do this is that then you when you find, for example, the association between exposure to increased heat index and the appearance of skin aging signs, then you add in UV exposure. And if it would be completely due to the confounding, then the significance would be gone. But what we found is that the effect was getting smaller, but it still remained significant. And this and some other controls clearly established that heat index per se is an environmental factor that likely contributes to skin aging in humans.
Well, this is bad news for those of us who live in a warm climate like I do in Austin, Texas, where it's still very, very warm, even in September and sometimes October. Okay, so we learned from this study looking at 3 different cities in India. And I remember very clearly during COVID when the pollution really dissipated because people were not out and about. You saw those amazing photos of the Taj Mahal, for example, where it was, on a normal day, very hazy and difficult to see. And then during COVID, it was absolutely clear. So I can understand the effect of pollution, especially in that country. Were the heat indices different in the 3 different cities, or do they have about the same heat index?
No, they also show differences in the heat index. And
Okay.
this is also the power of a study like this. So ideally, you always pick cities at different geographical locations, which give you a nice, strong gradient as it concerns the environmental factors you're interested in.
Okay. And so taking into account the UV exposure, as you said, if you look at the different heat indices, you could see different amounts of melanin production, therefore, in the different cities?
Well, we could see differences in hyperpigmentation. So, for example, pigment spots in the face were just more frequent when the heat index went up. What I should tell you is what we are looking at in this type of epidemiological analysis are really chronic effects. So we looked at the effect of an increase in ambient temperature over a 5-year period. Yeah. And so this is very different from, for example, experimental settings where you just quickly and strongly heat up skin, and then you induce a heat shock response. So these are really chronic effects, and already an increase by 1 or 1.5 degrees Celsius already give you strong effects when it comes to the development of, for example, pigment spots.
And one could assume, and obviously in this epidemiological study, it was much easier to assess pigmentation versus any other of the skin aging metrics. But one could assume that if you see an increase in pigmentation, then molecularly you would, at the DNA level, you would likely see changes related to skin aging as well. Is that correct?
What we saw is also that some of the wrinkle types in the face were also associated with an increased heat index. So I don't think it's specific for pigmentation. Yeah. It's probably associated with several other skin aging phenotypes. But as you said, also for the purpose of this study, of course, the hyperpigmented lesions are the easiest to quantify and to measure.
Right. So you took that finding and then you applied it to your lab, right? You went into in vitro data and research. Can you review that for us?
Yeah. You see, the epidemiology is always very powerful. And what I like about epidemiology is this is the way to really find out if there is truly a problem to human health or not. And this was a strong indication that there might be a problem for skin health. However, this could also just be by coincidence, because epidemiological studies can only look at associations, and this does not necessarily mean that there is a cause-effect relationship. So there are nice other examples where 2 things look very strongly associated, but from a mechanistic point of view, they don't make any sense, and then you have to question this. So we thought it would be very nice to also have supportive mechanistic data. And then you need to develop a model, and the model should be as close as possible to the real situation. But of course, it can never mimic the real exposure situation, because we cannot wait for 5 years to see the results in the laboratory.
Nor can you account for every variable, diet, genetics, stress level, all of it. So granted, you can't account for everything.
So what we did is first of all, we took normal human skin. This is this was leftover skin from plastic surgery.
Abdominoplasties or facial surgery?
These were abdominal plastics.
Okay, so abdominal skin.
For belly skin. Then this can be nicely cultured. So you have what we call an ex vivo human skin model.
Mhm.
And then we thought, well, relative humidity should be high. So this is easy to do because usually in an incubator, you already have a relative high humidity. But then we discussed how to mimic the slight increase in skin temperature. And normal skin temperature is actually only 31 degrees. So it's below our body
Core
temperature.
temperature. Yeah.
And when we just cultivated skin models at 31 degrees for up to 2 weeks, there was absolutely no increase in constitutive skin pigmentation. Already by increasing by 1 or 2 degrees, you could really nicely see a time and dose dependent increase in skin pigmentation. And this was the maximal if we went up to something like 34, 35 degrees. So we had a nice model.
Mm-hmm.
When you have a model, then of course you can ask many more questions. In particular, how does it work? What is the underlying mechanism?
Okay. And so, just to summarize then, we normally think about pollution affecting the skin as UV or ultraviolet pollution, as you said, microparticle pollution, diesel exhaust pollution, and that's part of the exposome, which is the external and internal factors that affect the skin in a negative way in terms of, contributing to skin aging. But now we have to add heat.
Mm-hmm.
And you've shown that very nicely, both epidemiologically in the study in India in 3 different cities, and now ex vivo, you've shown it as well with a slight increase over time of the temperature leading to increased melanin production in this abdominoplasty derived skin. What can we do to prevent that, Dr. Krutmann? Please give us some hope.
Yeah, exactly. Yeah. And don't deliver only bad news here. Actually, we found that this increase in pigmentation was always associated or went along with an increase in oxidative damage in the skin. So, for example, when we looked at the DNA of the skin cells, we found that in the situation where they were cultivated at increased temperatures, there was more oxidative DNA damage. And we also looked at the lipids and we looked at the proteins, and they were also more oxidized as compared to skin that was kept at normal temperature.
Okay.
And then, of course, the hypothesis was that very similar to what we already learned from air pollution, which also through oxidative stress is increasing skin pigmentation, the heat might use a similar mechanism. So then how to prove it? And the best proof would be that one treats the skin with an antioxidant and then heats it up. And if this no longer leads to an increase in skin pigmentation, then this would definitely prove that the oxidative stress is mediating the increase in skin pigmentation. Yeah.
Okay. So it's interesting. So you're saying then that heat leads to damage through the reactive oxygen species pathway, right? Through oxidative stress, essentially.
That was the hypothesis. And,
That was the
the
hypothesis.
way we tested this was that I asked my postdoc to go to the next pharmacy to buy a cosmetic product which, as active ingredients, only contains antioxidants.
Which is important, right? Because so many products are what I call Swiss Army knife products. They tend to
they have everything. They also
everything.
Have agents to bleach the skin, and so you never know when you have the net effect which ingredients are really responsible for it. So we wanted to have a puristic approach to be really sure that this is either mediated or not through oxidative stress.
This episode is brought to you by SkinCeuticals, the #1 medical aesthetic skincare brand worldwide and the leader in antioxidant science. To learn more, visit www.skinceuticals.com.
So let's take a pathway off to the side here and talk about that. So the product that your postdoc found was a CE ferulic from SkinCeuticals, right?
Yeah, actually, we like this product a lot because it's a very nice tool to really ask to address the question I was just asking, because it can be topically applied to human skin. It's because it is a cosmetic product. It is very well tolerated by human skin. And because the 3 active ingredients are all antioxidants. It's vitamin C, it's vitamin E, and it's ferulic acid. And we are very familiar with this. Actually, me personally, for a very long time, because my relationship to this company goes back to a personal relationship to the founder. And we did some very early experiments with CEF decades ago. So it's a very pure antioxidant containing serum, and we use this then to topically treat the ex vivo human skin models. And when we do this, we found that it can significantly and almost completely prevent the temperature induced pigmentation response.
And this was the experiment that you performed was over how many days?
This was always about 10 days total.
10 days. So a slow increase in temperature over 10 days with a daily application of CE ferulic to that ex vivo skin.
It was every third day we applied the CE ferulic. This was already sufficient. So it really is very potent strategy.
Interesting. Why do you think a daily application was not necessary and every third days was fine?
Let me say it this way. We made our life a little bit more easy by just applying something from the outside under sterile conditions every third day. Because whenever you take the lid off and you take the skin out of the incubator, there's a risk that you might induce some contaminations. So we tried to keep this risk as low as possible.
Certainly, in daily practice, though, we recommend the application of CE ferulic or an antioxidant serum or gel on every day, because it by itself, once you apply it, it gets oxidized as well. It has to deal with a lot of factors related to the environment. So probably a daily application, in vivo is the right way to go.
Definitely, and you should do it in the morning before you leave home.
Mm-hmm. Yeah. Would you recommend it before sunscreen or after sunscreen?
That's a little bit of psychological. It's a philosophical question, but usually I tell my patients to apply before the sunscreen and finish with the sunscreen.
Yes, I do too. Okay. From thinnest to thickest is how we think about things in terms of applying it. Also, it makes it easy for patients to remember.
Exactly.
Okay. All right, great. So the application of the CE ferulic serum every third day to this ex vivo skin led to essentially negated any heat-related effect on melanin production, therefore.
So, I mean, then of course we are always interested in getting the best possible publication, and this usually requires that when you do an experiment like this, it should also be vehicle controlled.
Okay.
Yeah, because you theoretically, although very unlikely, you cannot exclude that there might be something in the galenic formulation that could possibly also interfere with the heat induced pigmentation. And this was the time actually when we called up SkinCeuticals, because I needed the vehicle, that is the product without the active ingredients. And then they kindly agreed to scientific collaboration, and they provided us with exactly the vehicle the CEF is usually being kept in. And the vehicle alone had no effect. So now we have very nice data. So it's a vehicle controlled ex vivo study to make our point.
Great data. So in terms of the amount of heat required or the increase in temperature required to produce any effect on melanin production, what was the minimum amount that you found?
Actually, we already see a slight increase. If we just increased by 1 degree, and it just was dose dependent. So with 2 degrees, it was stronger. With 3, 4, we never went up above 37 degrees because we wanted to stay away from a heat shock response. And this is physiologically relevant. So there are studies where people have, for example, measured skin temperature in people who are asked to sit in the sun. Actually at a swimming pool, probably a very nice experiment for the volunteers. and they found that just exposure to sunlight for 1 hour brings up the average skin temperature by 1.5 degrees Celsius. So this is actually the range we are also dealing with. And
this is so applicable to our daily practice, and we don't think about this, but especially in a climate which is warm but perhaps not sunny. Of course, we ask our patients to wear sunscreen because of the UVA getting through the clouds. That's well known. But just the warmth itself now is going to really change our practice and lead to the greater use of an antioxidant serum in the morning, because we need to, as part of the overall exposome, we have to include that. Warmth is now added to, as we mentioned, diesel exhaust and particulate matter and air. All of it now is something where we know from lots of data, Dr. Krutmann, because I know you've been involved in this as well, along with Dr. Valaki and others, that so much of what we get exposed to living in urban environments can be harmful to the skin, but we thankfully have the ability to use these topical antioxidants to really prevent and preclude their harmful effects on the skin. Is that right?
That's absolutely true.
Yeah.
There are, of course, some very important questions that also need to be addressed. And in the answers, not only the dermatologists, but also the consumers and the patients will be very interested. One is, of course, what is the relative contribution of each of these factors to, let's say, lentigo solaris, which, as we know now, is a misnomer, in fact. So an age spot.
Yeah.
And this is something that we are currently tackling. It's not trivial. You need to have very sophisticated biostatistical models to be able to address this, but it's possible. And we just published a proof of principle study where we asked this question for age spots in elderly German women, so almost exclusively Caucasian women. And there you are really able to establish a hierarchy. So no surprise, UV exposure was the most important exposomal factor. But then already at position 2, and with a very slight difference, air pollution came in. And for example, tobacco smoke exposure was of almost no relevance. And when you do the same analysis for wrinkle formation, you get a different sequence. Then UV is still the most important, but then the second most important is tobacco smoke exposure, and air pollution is much less relevant. And this just shows you that we are really now able to talk about relative contributions. And these relative contributions will be different very much depending on where I'm based, how the exposomal conditions will be for a specific day. And of course, they also depend on my genetic makeup, because it's always the interaction between the exposomal factors and the genetic factors. And the whole idea is that the more we learn about this and the better our models will be. And this, of course, is also a question of how big the data sets are that we can feed into the models. The closer we will really get to science-based development of targeted cosmetic products to really provide perfect protection of consumers against environmental threats.
Well, this opens up a whole nother line of questioning, right? So in terms of personalized skincare, like you said, some of it is related to environment, some of it is related to genetics. Have you started looking at skin scrapings or even skin biopsy models from different genetically different people in different parts of the world, what those genetic profiles look like and how that could dictate the different ingredients, application strategy, concentrations of ingredients, formulations, all of it related to personalized skincare? Is that something that's happening right now?
Yeah, I mean, we are still more in a cruder state, so I would not really talk about personalized, but at least we can now start talking about targeted to, let's say, susceptible groups.
Okay.
So a very good example, I think, is phototypes. And just think of ultraviolet radiation exposure. The darker your phototype is, so if you're phototype 4 or 5 or 6, then you need much better protection against UVA radiation. And you need less protection against UVB because you have more eumelanin already around the nuclei of your keratinocytes, so you're better protected against skin cancer. And it's the other way around for the lighter skin types. This is already a good example. So you should not give the same sunscreen to everybody. Yeah, at least you need to take into account the phototypes. And we would like to reach at least a similar level now also for air pollution and for the heat index problem, because we believe that there will also be single nucleotide polymorphisms which might define the susceptibility towards air pollution and towards heat-induced skin aging phenotypes. And this is actually ongoing. And we also have some industry help, for example, from SkinCeuticals to do this. there are two strategies. You can do this unbiased, and it's called a GWAS analysis, and then you look for gene-environment interaction. This is very nice, especially because you might find things you haven't thought of before, but the problem is it requires very, very high numbers of volunteers. So here we are always talking 6-digit numbers. So these are also very expensive studies and take a lot of efforts, but it's possible. And the other possibility is that you come up with an educated guess, then you look just targeted and you say, oh, maybe this gene is interesting. And then you just compare SNPs in this gene, and then you see if this defines susceptibility or not. But this is something that keeps us very busy.
Interesting. And do you do any research on systemic antioxidant exposure? So, for example, we've talked about topically applied antioxidants and their effect to minimize the effect of the exposome. What about those that are taken ingested?
Yeah, excellent question. And in particular, because we know that when you orally take antioxidants, a lot of them ends up in the skin and will also define your resistance or your susceptibility towards environmental threats. And I personally believe that because we see some inter-individual variability, for example, in the pigmentation reaction towards topically applied air pollutants, that this is very much also influenced by dietary habits. So if you, just before we do the Düsseldorf pollution patch test, if you drink a lot of tomato juice and you have very high lycopene content, you might be better protected. And it's on the list of things that we would actually like to test. And there is some indirect evidence that would support that this is indeed the case.
Absolutely fascinating. Well, Dr. Krutmann, thank you so much for your time. I have learned so much now, and unfortunately now we need to be concerned about the heat that we are exposed to in addition to everything else. Luckily, though, as we've mentioned, that the topical application of an antioxidant serum seems to be protective against most of everything that has been studied thus far. There may be an opportunity to ingest antioxidants as well, as we just discussed, to enhance the protection against the negative effects of the exposome. But otherwise, absolutely fascinating. I'd love to, in maybe a year or two, come back and talk more about your research. I know it's forever changing and expanding, and you're building upon what you've done in the past. So I hope you'd be open to that.
Sure. Thank you very much, Ted, and thank you for the interest in our work. I greatly appreciate this.
Oh, wonderful. So this is Dr. Ted Lane. I am the host for Skincare Confidential today with Dr. Jean Krutmann. Please subscribe, give us a like, and refer us to a friend. Otherwise, we'll see you at the Science of Skincare Summit in November. Thank you.